Good afternoon, my name is Maggie. I'm an intern in the Rediske lab, and this summer I spent my time doing an investigation of the Escherichia coli contamination of Little Black Creek in Muskegon. So for the past 2 summers, I have had the privilege of working on the beach monitoring team. So we monitor Lake Michigan and inland lakes in the general West Michigan area. We take the water samples back to our lab to test for E. coli, but we also do a bunch of other routine surveillance as well that's available to the public. So basically last year, one of our beaches, which is at the PJ Hoffmaster State Park Campground, was closed due to erosion and debris. So they moved the beach a little bit farther north, and that was where Little Black Creek, which is the subject of my presentation, was discharging into Lake Michigan. And since it is a creek, it had E. coli contamination, which was raising the E. coli counts of Lake Michigan at that area, which was cause for concern. And so then we started investigating the creek itself, and the contamination was further concerning. So we decided to take a closer look at the contamination of the creek. So here are the sites that we monitored in 2020 on the left, and then in 2021 on the right. So last summer, we monitored all along the stretches of the creek. We started where it leaves Little Black Lake, that's site 1. I believe so much here. So site 1, and then here in the middle of the PJ Hoffmaster State Park, and then site 3, which is the one that I mentioned earlier, is at that beach where it's emptying into Lake Michigan. Site 4 is a really deep site along one of the trails in the campground. Site 5 is at the entrance to the campground, and then site 6 is this separate branch over here that feeds in right between sites 4 and 5. This is a mainly residential area off of Lake Harbor Road. So in 2021, this summer, we focused mainly on sites within the campground. We kept site 6 because it was notoriously high last year, and then we kept monitoring site 3 as well because it's at the beach. We added site 7, which is a mixing site between the creek water and the Lake Michigan water to monitor how much the creek water is affecting the quality of the Lake Michigan water. And then we also added this site here, which is site 8. There's a little bridge over the creek that connects the 2 halves of the campground, and kids are always playing in this area, so we decided to monitor that location as well. So here's some pictures of myself and my partners monitoring the creek and the Lake Michigan water. So one of the things that we do with the water when we bring it back to the lab is KOHLER. So a KOHLER test takes 100 mils of the water sample and mixes it with mug mix. And there's the full name of the mug mix if you are interested. But basically it has a fluorogenic compound that binds specifically to an E. coli enzyme, and that makes it glow. So after you mix it up with the mug mix, it goes in one of these trays over here, incubates for 18 hours, and so then we can read it the next morning. As you can see here, some of these wells are yellow, but they're not glowing, and then these ones are very clearly glowing. So you count the wells that glow, and then that will give you an MPN, or most probable number of E. coli in that sample. And that's reported in CFU per 100 mil, and so that's the number of colony-forming units per 100 milliliters of water. And so this picture shows a partially fluorescent plate, a fully fluorescent plate, and then this is a lab blank, so it's not fluorescing at all. What we do though is if we take a left, center, and right sample from the creek, we'll calculate a geometric mean of those things. If that geometric mean MPN is above 300 CFU per 100 mil, that would be a total body contact hazard, which means you don't want to be swimming in that water. And then if it were to be above 1,000 CFU per 100 mil, that would be a partial body contact hazard, which means that any contact with the water, even in an activity like kayaking, for example, would potentially be a health hazard. So here are the KOHLER results from 2020. As you can see, many of the results are exceeding that partial body contact limit in sites 3, 4, and 5. And then this red line here marks about 300 CFU, which is the full body contact limit. And almost all of the results exceed that limit as well. So pretty much all of the results that are above this line, that's like hazardous water to be in, and probably not good that there are children playing in that creek. The upper limit of quantification that we have for the KOHLER test is 2,419.6 CFU per 100 mil. And that was reached in sites 3, 4, and 5 on this green day and then the pink day as well. But as you can see, for example, the green day is July 29th. We did go out and sample again that next day to kind of monitor the contamination. And then, like, as you can see, for example, at site 3, it did come down that following day, but that's just something to make note of. Here's the results for 2021 sampling. We're still in the middle of this sampling period. We have one more sampling week next week, but this is the data that we have so far, and it is considerably lower than it was last year, which is a good thing. I included the 300 line again, and so we do have several events that are above the 300 CFU. So again, the water there's still contamination of the water, but it is significantly less than it was last year. As you can see, there's nothing that even exceeds that 1,000 level. And we're not really sure why that is, but we do have further tests that we're conducting. And so the way that we're doing that is we're using polymerase chain reaction, or PCR. And so PCR is used to measure the DNA in a sample, not just the live culturable cells that KOHLER measures. So it's like a more accurate depiction of how much E. coli is in the water. And so after we do the KOHLER test, we take 100 more mils of the water sample and we filter it. And then basically there is like a long drawn out method to extract a clarified extract from that filter. And then that goes in this plate right here along with the master mix. And then we can do qPCR, which is quantitative polymerase chain reaction, which amplifies the DNA that's already there. And that uses fluorescence to measure the amplified DNA, and it measures when it crosses a certain threshold, and that gives us our value for how much E. coli is in there. That was used to test for the human fecal marker in 2020, and that's known as microbial source tracking, to try and figure out where the contamination is coming from. But in 2021, we have available to us the digital droplet PCR, or ddPCR. And so this is similar to qPCR, except in the fact that it uses droplets. So we have an automated droplet generator that will make a bunch of little bubbles and droplets in this plate. And then the droplets are assessed for fluorescence. And so because it's generating all of those droplets, it's a better method for samples that have low concentrations of the target. And so in 2021, the digital droplet will be used to run MST. So in 2020, like I said, we tested the water for the human fecal marker HF183, and all of the sample sites were negative. So that means that that was not the source of the contamination. And unfortunately, that left us at like a blank. We didn't really know what it was. So coming back this year, we decided to test it for a variety of markers for some of the animals that are a little bit more prevalent in that Hawkmaster Campground area. And we have not completed these tests yet because, like I said, we still have one more sampling event next week. Once we have all of our samples ready to go, then we can start running the MST to try and figure out where the contamination is coming from. And we will be running the human marker again, a gull, dog, and a ruminant marker to see if we can get a positive for one of those, and then that will let us know like what the source of the contamination is. Fecal contamination of any kind, regardless of where it comes from, can be a health hazard for those who come in contact with the water, and so it would just make it easier for us to address the issue if we can figure out where that contamination is coming from, which is why MST is so helpful, because it lets us test for specific species. So I have a couple of thank yous. I wanted to thank Dr. Rediske for giving me so many opportunities and entrusting me with the responsibilities of the projects that I've been privileged to be on. And then also thank you to Brian and Molly, who's not here, but they've trained me so well and just made like an awesome lab environment. And I really love what I do. And then all of my beach monitoring partners have just been a joy to work with. And the rest of the AWR staff too. There's just a great environment here, and I really enjoy being here. So thank you all for your time.
Are there any questions for Maggie?
Now, do you feel pretty confident that the source is in the park itself, though, regardless of what the animal source was?
Yeah, so the sites that we measured in 2020 were kind of all over the creek, and so for example, like 1 and 2 were just kind of consistently lower than all of the rest of them, especially site 1. Like if I go back to the results, which is why we kind of decided to focus on the campground itself, because all of the sites that are in the campground were significantly higher than site 1. The only thing to note though is that site 6 is kind of off by itself, and site 6 was our 3rd highest site in 2020, and it is our highest site this year.
That's
So
what I was concerned about. I mean, that's a drain, and so I'm just curious if there might have been sources coming out of it. I don't know if those homes back in there are on septic or if they're on sewer. Do you know that? Okay.
I don't know.
Thank you.
I just had a question, it's similar to Al's train of thought there. If there's any chance or thought that it could be agricultural, like animal related type.
That's
things
kind
going
of
on.
that's the thought process for bringing in some of those other markers that we're going to be testing for. This area, like this specific area, isn't too agricultural. A lot of it is residential and state park. I think that's pretty much the only thing that's in this area. But if you move out a little bit, then there's definitely a lot more of like the agricultural areas. But that wasn't that wasn't the direction that we decided to go in first. So we'll see how the deer and the seagull and the dog markers come back, and then perhaps we'll broaden it to like the agricultural like cow and pig markers as well. So
Can you share the data with DNR in the park?
we were in contact with DNR last year. They kind of helped us. They had septic. They had a septic tank that they had replaced in 2019, and so that's why we tested it for the human marker. But obviously, it came back negative. But we have been working with them. I don't believe that we reported our data for this year to them though, because we're still collecting. So.
Any further questions? Okay, well, hang on.